have this other, there's a whole box in the textbook that talks about the fact that there's not just a mitochondrial form of this enzyme, but there's also a cytosolic form. And the cytosolic form is involved, it does the same reaction in the cytosol, but it does that as part of fatty acid metabolism. So that's not a part of fatty acid metabolism that we need to study. But what's interesting about it is that the cytosolic isoform also has an FES cluster, and it's regulated, that enzyme is regulated in a really interesting way. If there's not enough iron around, that enzyme won't work because it won't have an iron sulfur cluster. There won't be enough iron to assemble the iron sulfur clusters, right? So the APO enzyme that doesn't have an iron sulfur cluster is able to bind the messenger RNA, which has been transported from the nucleus into the cytoplasm. This is a cytoplasmic lysozyme. So you have an enzyme that's basically doing something metabolic. And now, with an iron sulfur cluster in it, if you take the iron sulfur cluster out of it, because there's not enough iron to make that sulfur cluster to begin with, now this new protein, which is the same protein, but it doesn't have the iron sulfur anymore, it's now able to bind a messenger RNA molecule. So this is an RNA binding protein, right? And so, which messenger RNA of all the thousands in the cell do you think it binds? It binds a messenger RNA for a protein that takes up iron from the environment, right? And when it does that, it helps that messenger RNA not get degraded. So one of the mechanisms for gene expression, for controlling when genes are on and off and how long they're on for, is you got to make a messenger RNA out of them to make protein. The messenger RNA is, on the ribosome, but it's inherently labile. It's an RNA molecule. Doesn't have much structure to it intrinsically, so it'll fall apart. If you can find a way to stabilize it, then you're ultimately going to make more protein from it. So if there's not enough iron around, you bind that messenger RNA. That messenger RNA makes, on the ribosome, makes more iron uptake protein. So it's a way to reconstitute the iron in the cell. Everybody see that? So you can look at the box in the textbook if you want more information about that. It's such a neat mechanism. It's an example of moonlighting for enzymes. So normally we think, well, enzymes have one function, that's it, right? And then we're done, we know everything about them. But increasingly that's not true, right? In this case, get rid of the cofactor, all of a sudden it does something else. And there's many more and more examples of enzymes that you think you know what they do. Amino-acyl tRNA synthetases are the enzymes that translate the genetic code. And we thought, well, that's what they do, and we don't have to think about it anymore. Then you start to find out that in eukaryotes, these enzymes get big domains added to them. Then you start to recognize that they're involved in processes like signal transduction and cellular differentiation. So the cell has these big proteins around. Why not, rather than making a whole new one from a different new gene, let's just co-opt this guy and have him do something else. As long as we can still get the old function satisfied, why not?
So if it's bound, and I get that it doesn't have complete processivity, but if it's bound, how is it being translated?
Well, it's not translated all the time. So to the extent that the message is free, maybe the ribosome's moving along the message, and you have this whole free part that might get eaten up or degraded chemically, but not if apoaconitase binds it. So something like that. Good question, though.
What does the APO stand for?
APO means without a ligand or without a cofactor. Like it means empty.
Okay.
APO means empty. So in this case, aconitase is empty if he doesn't have his iron sulfur cluster to make him happy and allow him to bind citrate well. But he found a way to be happy without the iron sulfur cluster. Go grab the messenger RNA. What's particularly neat is it's not just any messenger, right? I mean, there's like thousands of messengers. It's the message for iron uptake protein, right? So what's up with that? Good question to take into the next term, those of you going on. Okay. When the citrate binds to aconitase, you'll note that citrate has two groups on it. It's not a chiral center, but we call this prochiral. it's got two, it's a potential to become chiral. It's got this A1 and A2 on citrate of the same group, and this is getting reacted by. So it matters as far as the positioning with respect to the iron sulfur cluster center, which way this molecule is oriented. So if you consider this carbon is tetrahedral, right, the CD binding sites at 109 degrees are going to work a certain way, so that this OH is coming out here and is positioned next to the iron sulfur cluster. Right? So the asymmetry of the enzyme surface means that CD only binds a certain way, and therefore that A1 is in here, and so you can react off this side. Does that make sense? That's a way to distinguish the two CH2 COO minus arms of citrate. Okay. The next two steps are oxidative decarboxylations. Here it says NADP, but this P is in parentheses to indicate that it could be NAD or NADP. So for our purposes, we won't worry about the fact that there's an isozyme that can handle NADP. There's probably a little bit of NADP around in the mitochondria. Not a lot, but some, and so why waste it? So there's an enzyme that allows use of it. CO2 is split off, and we have a 5-carbon compound, and then here we have the 5-carbon compound incorrectly drawn on the slide. Not my fault. That's what the Leninger gave me. It's a slide here, but it's actually printed properly in the book. So alpha ketoglutarate, which is drawn correctly here and not reproduced correctly here, is another CO2 is split off. In both of these cases, we've got the formation of an NADH. Right? Both of them are also strongly favored in the direction of splitting off CO2. CO2 is a very low energy, happy, stable molecule. You can form CO2, you're almost always going to have a nice negative delta G in favor of forming it. Right? So these are irreversible steps that drive the cycle forward. So we have these two oxidative decarboxylations. Trying to decide if I should talk about the mechanism now or later. I think I'll talk about it later. What I want to talk about in terms of this mechanism is the fact that this oxidative decarboxylation is breaking a carbon-carbon bond, but there's no shift base like in aldolase. There's no thymine pyrophosphate like in transketolase, which are the two mechanisms we learned to break a carbon-carbon bond. So if you go back to chapter 13, I think it's page 513, it talks in detail about how you cut carbon-carbon bonds. There's actually another mechanism mentioned that we haven't talked about yet, and that's how this enzyme works. In fact, both of these enzymes work. So we'll talk about that next time, but you might want to take a look and see. There's 3 different strategies to know about when you talk about breaking carbon-carbon bonds. But even though this is a decarboxylation, very stable thermodynamically, we're talking about the kinetics. We're talking about how fast can you cut it, right? How much can you bring the transition state down? It's still a carbon-carbon bond getting broken. So something special has to happen. So the second of these enzymes, alpha ketoglutarate dehydrogenase complex, functions the way the PDH complex does. Pyruvate dehydrogenase complex. So I misspoke. Pyruvate dehydrogenase complex we talked about at the beginning of class. This complex here in the citric acid cycle. Here's another complex in amino acid metabolism. There are all these big particles. It's the same. You can see it's the same cofactors in and out. So I misspoke. This one does not work by that third mechanism I was alluding to. This one does. So this one does not. This one has TPP in it, but TPP is in PDH, and it's also in this. This one does not have TPP or shift base. So the last steps, we have this 3-step sequence, right? Reduction, hydration, and reduction. And I'll close by talking about this succinyl-CoA